Welcome back to part two, AP Bio, and I want to hear what your thoughts were on this question. So, which part of a generalized invading pathogen will be recognized by a macrophage, and what is the structure called on COVID-19? Do you remember what the vocabulary word is? It's called an antigen. Antigen. And on COVID-19, the antigen for COVID-19 is the spike protein. This is going to be important for understanding how scientists developed vaccines in a minute. So let's talk about how COVID-19 infects your cells. Now, this is going to be fairly complicated. So in the lecture guide, I have a half sheet printout version of this diagram, and I encourage you to follow along using this diagram. If you are in my class in person, we glued this diagram into our notes, and I encourage you to color in this diagram because as we go through it, it's going to get kind of complicated and messy. So I want to just make sure you're paying attention to the different steps. So the first thing that happens when COVID-19 enters your body, and we know that it is a respiratory illness, so it infects via your respiratory system. So it's going to infect via your mouth and nose. It gets into your lungs and it's going to attach to the cells in your lungs, which is why you typically get a sore throat, you might kind of cough, and you might have trouble with shortness of breath because the virus is attaching to the cells in your lungs. The way it does this is COVID-19 actually uses a receptor on your cells called the ACE2 receptor. And it actually manipulates this receptor to allow it to infect your cells. And so this allows the virus to be endocytosed into your epithelial cells in your lungs. Now, before we get too deep into this mechanism, why is there a receptor for a deadly virus on your cells? Why is the ACE2 receptor even there? We wouldn't think that we should have a receptor for a virus on our cells. Well, let's take a look at what the ACE2 receptor normally does. So these are the reference images I pulled from. I actually drew my own diagram that you can draw in the lecture guide for yourself, okay? And we're going to start by talking about symptoms. So the ACE2 receptor normally in your cells is involved in a pathway that helps with blood pressure. So if your body receives the symptoms of low blood pressure or low sodium in your kidneys or even stress, your body starts to produce this molecule called angiotensinogen. Angiotensinogen. And angiotensinogen is a hormone. Now, angiotensinogen is going to be involved in the following signal transduction pathway. So if you're drawing this in your notes, I encourage you to draw this structure out. It should take about a full page. There are 5 different proteins that we're going to be paying attention to. We have the ACE protein, signal transduction pathway 1. These are just generalizations. A lot of what I'm doing here is condensing a lot of different pathways. So I've generalized this diagram to be more specific to AP Bio. We have the ACE2 receptor. This is what COVID-19 is eventually going to bind to. And then we have the G protein coupled receptor, which if you don't remember G protein coupled receptors, I encourage you to go back to that section in signal transduction pathways and to review that pathway, okay? Now, if we go back, we are looking at normal ACE2 reception. We have angiotensinogen being produced because of low blood pressure or stress or low sodium. So that's what this black molecule here is going to represent, okay? It's going to produce angiotensinogen, and angiotensinogen is going to be taken by the enzyme renin. And renin is going to convert it into a new structure called angiotensin 1. Again, this is a great example of scientists naming things very confusingly, but it's because they're very closely related structures. Angiotensinogen, this black circle here that I've drawn, is very similar to angiotensin. They've just been slightly altered by this enzyme renin. Now, angiotensin 1 is going to be absorbed and enzymatically react with this ACE receptor. ACE is an enzyme that will convert angiotensin 1, or ANG1, we'll abbreviate that as ANG1, It's going to convert it into Ang2 or angiotensin 2, which is very similar, but slightly different than angiotensin 1. Now, angiotensin 2 is going to trigger two different signal transduction mechanisms. And those transduction mechanisms are going to produce the following response. They're going to increase your blood pressure. They're going to increase inflammation in light of a potential infection. They're going to cause vasoconstriction. So they cause your blood vessels, blood vessels, to get smaller. And in the event that they cannot get rid of whatever it is that's causing these things, they may cause cell death, right? This is the idea of, okay, if this cell is broken beyond repair, we should get rid of this cell so it doesn't proliferate and create more broken cells. Now, we don't want this to happen for an extended period of time. Increases in blood pressure and increases in inflammation and vasoconstriction and cell death are not good for you. You don't want your body to be in a constant state of these processes, which is why constant stress can happen in certain traumatic circumstances, or if you're, a highly ambitious AP Bio student, it can cause a lot of really significant issues. Now, luckily, your body has a mechanism to adapt to this. As your body starts to produce more angiotensin, it's also going to trigger a separate set of signal transduction pathways. So I've labeled these STPA and STPB. I apologize for the blurry picture there. So STPA and STPB, they are going to trigger this G protein coupled receptor. And they are going to trigger a separate response that will lower your blood pressure, decrease cell proliferation, so it'll stop cells from going through cell division. It will kind of protect the cells. The cells will kind of go into defense mode rather than investment mode. So they're not going to be doing really energetically costly things. They're going to be doing things that maintain the cell's integrity. And it'll cause your blood vessels to dilate, so to get bigger. And in this way, you have a feedback loop where one response increases blood pressure, one response decreases blood pressure. And so you end up with a balancing mechanism for any of these stress responses that are produced from angiotensinogen. Now, where does ACE2 come into all of this? Where does the ACE2 receptor get involved? Well, angiotensin 2 gets attached to the ACE2 receptor. And the ACE2 receptor then creates a precursor molecule that emphasizes STPA and STPB. It triggers a reinforcement pathway that will continue to lower blood pressure, continue to lower cell proliferation, or increase cell proliferation, and increase this cell protection mechanism, right? So these mechanisms are a feedback loop that feed on each other and balance each other out in the ideal circumstances. Now, one thing that I want to point out, I believe I said cell proliferation is inhibited, but cell proliferation is actually increased. So after your body recognizes that it's no longer in a stress mode, it's no longer in this sort of, okay, we need to get rid of these broken cells, it'll cause those cells to actually proliferate. So it'll cause your body to reproduce those cells that have gone through cell death. So I think I misspoke a second ago. I just wanted to emphasize that the second response in this pathway, cell proliferation, is an increase in cell proliferation, not a decrease in cell proliferation. If you have any questions about that, feel free to put those questions in the comments if that was at all confusing. Now, what happens when COVID-19 attaches to the ACE2 receptor? How does this pathway become manipulated and changed? Well, if you think about this pathway, if you work backwards, COVID-19 blocking the ACE2 receptor means that it is inhibiting the reinforcement of these signal transduction pathways that lower blood pressure, lower cell proliferation, and increase vasodilation. So when COVID-19 bonds to the ACE2 receptor, you basically get a continued increase of blood pressure, inflammation, vasoconstriction, and cell death, which can be really dangerous if it's allowed to be continued. So this is sort of a summary of how that process works. If you want to write down these notes, this is just a summary of each of the different steps that I just talked about in this diagram. Now, if you think about this, this is why people who have high blood pressure are very susceptible to more extensive COVID-19 symptoms. Because if you already have high blood pressure and then you get an increase in blood pressure on top of your already high blood pressure, that's going to be incredibly damaging and dangerous. So this is why you hear about some of those symptoms regarding the COVID-19 infection. Now, let's go back to the mechanism of infection. So the COVID-19 virus bonds to the ACE2 receptors in your human lung cells. Now, where do you have a high number of ACE2 receptors? In your lung cells. So this is why people typically have respiratory symptoms. Now, individuals with high blood pressure, they will get this compounding effect when infected with COVID-19, right? We just talked about that. Now, after COVID-19 gets into your cells, it is going to release the RNA of COVID-19 into your cell. And that is going to cause the cell to basically be hijacked into producing viral proteins instead of cellular proteins. Now, if you're not sure how this works, I encourage you to go back and review protein synthesis. Look at the central dogma. This is something you should have learned in general biology, but go back and look at that mechanism because it's going to help you understand how the COVID-19 RNA is able to be manipulated and used by your own cells. So in light of that review, I encourage you to answer this question. Which organelle is going to be used by the virus to make a protein? There's a little bit of review from organelles in general biology. That's going to be the ribosome, okay? Now, once the virus has made, or the viral RNA has been translated into proteins, step 5 is going to be the assembly of that virus and then the release of that virus out into the extracellular space. This is a video that shows that process. So before I play the rest of this video, I'm not actually going to show you guys the whole video. I just want to show you the ways that COVID-19 gets into your cells. But I encourage you to go back and watch this video for the rest of the explanation of viruses and things like that when we're done with this lesson. Okay, so I encourage you to go back and watch that video if you're curious about those additional pneumonia symptoms. For now, we're going to go back and consider how the COVID-19 infection caused us to develop certain symptoms and then talk about the vaccine. Okay. So, how does your immune system respond to getting a COVID-19 infection? Well, once a macrophage recognizes that a COVID-19 molecule is in your cells, it's going to consume that COVID-19 virus and then display the viral antigens on its surface. The problem with this is it usually already happens after the virus has infected your cells. And so you've already got an infection before the macrophage can do anything about it. Now, the macrophage will then recruit helper T cells, and the helper T cells will recruit cytotoxic T cells and B cells to then go and make additional antibodies in response to the infection. It'll also create memory cells so that the next time you are infected with COVID-19, your body recognizes that infection. The problem is spike proteins keep mutating. So every time you reintegrate a COVID-19 infection into your adaptive immune system, the infection itself changes. So your body has to keep adapting its response to that infection. Eventually, the cytotoxic killer T cells can get after the infected cells and kill them. Now, let's take a pause for a second and consider the following questions. Using what we have learned about ACE2 in the lungs, explain the following. Why would COVID-19 cause damage in the lungs? And why are people with high blood pressure at greater risk for having a strong COVID-19 response? Take a second to answer these two questions. Well, I'm hoping if you did answer those questions, you were able to consider that COVID-19 causes damage to the lungs because it is effectively reinforcing a cell death pathway. So go back to that diagram we drew in our notes and consider how COVID-19 causes increased cell death, specifically in the lungs. And additionally, people with high blood pressure are greater at risk for having strong COVID-19 response because COVID-19 increases your blood pressure already. And so you basically get an additional symptom on top of your previous symptoms. Now, let's talk about the vaccine. Scientists have had a difficult time of trying to trigger the immune response without hurting the organism, right? Without giving a person a terrible sickness. So how do we do that? How do scientists do that? Let's consider this question. Which parts of the virus could you use that are important for the immune system to have access to, but won't give the person the illness? I'll give you a hint. It's one of the three: phospholipids, proteins, or RNA. Which one of those three things would tell the immune system what to do, but not infect the person? If you said the spike proteins, you are correct. So what scientists did, they actually practiced with a couple of different types of vaccines. And this diagram shows how they made different nucleic acid vaccines. One mechanism they tried was using a DNA vaccine. But the problem with a DNA vaccine is that they had to use a process called electroporation, where they basically made the cell membrane kind of flexible, and that allowed the DNA to be absorbed into the cell. But it wasn't very efficient, and it caused a lot of mutations to the DNA. So instead, what they tried is an RNA vaccine. And this is the crazy part. What they did, they took phospholipids and they took the RNA of just the spike protein, not of anything else in the virus, just the spike protein. And they injected that. If you got the vaccine, they injected the phospholipids and the RNA into your cells. Now consider for a second, why would the vaccine need to have phospholipids? And I'll give you a hint, it's in this diagram. See how the phospholipids here merge with the cell membrane? Well, if they just gave you RNA, it wouldn't be able to fit through the cell membrane because it's not permeable enough to RNA. RNA is too big of a molecule to fit through the cell membrane. So they give it to you in a phospholipid capsule, and this allows it to get into your cells, produce viral spike proteins, and then your immune cells, aka your macrophages, can recognize those spike proteins and send an immune response, aka your helper T cells, to trigger an immune response without ever getting the infection. So this is how we were able to develop a vaccine, right? Now, why do you think the vaccine developers used RNA instead of DNA? Think about this for a second. I kind of already mentioned it, but why would you want to use RNA instead of DNA? Well, consider that question as you also move through this diagram, right? If we were to use a DNA vaccine, there are a lot of additional steps that we would have to use, and it makes it more challenging to mass produce a vaccine. Now, let's consider one final question. And if you want to learn more about this development of the vaccine, I've linked this article here, which has some amazing graphics to help you out. Okay. And it talks about the immune system response as well. Now, to conclude this lesson on COVID-19 and signal transduction pathways, I want to leave you with this question. What might have caused different folks to have different responses to the vaccine and to COVID-19 in general? For example, I know a lot of folks that had neurological issues with COVID-19, and they didn't have as many respiratory issues. Now, there's no straightforward answer to this question. There's no real right answer to this question, but it's just a question to make you curious and consider how people might respond differently to different vaccines. With that, that concludes our lesson on signal transduction pathways and COVID-19 virus. I hope that was interesting and I hope it was helpful for you, and I'll see you in the next video.